Everyone, welcome back to my channel. In this session, we'll learn about one of the most important concepts in the field of biochemistry, that is electron transport chain. So in the field of biological oxidation, I mean to say when you consume the foodstuffs that consisting of carbohydrates, proteins and lipids. So once these things enters into our stomach, they get split. Since these are the complex molecules in the stomach, these things will get splits to give the simpler units. So that is carbohydrates on breakdown will give monosaccharides, and proteins on breakdown will give amino acids. Lipids on breakdown will produce fatty acids, and these are the energy rich compounds that will give energy for us when they undergo metabolism. That means these simpler molecules that are produced due to breakdown of this complex molecule will undergo the process of metabolism, and the metabolism of these reactions will actually takes place through various or series of reactions, and through that series of reaction these molecules will undergo metabolism, and the final products of all the reactions, final product of the reaction will be the two materials only, that is carbon dioxide and water. These are the ultimate products that are going to produce during the metabolism of this monosaccharides, amino acids and fatty acids. So here I said the metabolism of these components will take place through series of reactions, and among the series of reactions through which these materials will undergo metabolism, most of the reactions are dehydrogenation reactions. That means removal of hydrogen from the substrate molecule. So that means in dehydrogenation reaction from the substrate molecule that are involving in the biochemical reaction, from the substrate molecule one or two hydrogen atoms will get transferred from the substrate molecule to hydrogen acceptors. That means the components that are going to receive the hydrogen atoms from the substrate molecules. So during the dehydrogenation reaction of the metabolism, from the substrate molecule one or two hydrogen atoms will be transferred to hydrogen acceptors, and the hydrogen acceptors that comes into picture in the process of metabolism are preferably they are the specialized coenzymes, and these coenzymes will receive the hydrogen from the substrate molecule. And if you want to give the examples for the coenzymes that actually act as hydrogen acceptors in the process of dehydrogenation during the metabolism, the very simplest examples are NAD, which is called nicotinamide adenine dinucleotide. One more example for the hydrogen acceptor is FAD, called as flavin adenine dinucleotide. One more example for this is FMN, which is called flavin mononucleotide. This flavin mononucleotide is actually derived from vitamin B2, which is also called as riboflavin. I repeat, FMN is actually derived from the vitamin B12, which is nothing but riboflavin. And during the biological oxidation, these coenzymes will get reduced to change to the form of reduced coenzymes. Means during the oxidation, these materials will accept the hydrogen atoms from the substrate molecule and make the substrate molecule to undergo oxidation, and themselves they undergo reduction to form a reduced coenzymes. So NAD will get reduced to give NADH. FAD will get reduced to give FADH2. So give a careful observation to the reduced forms of the coenzymes. NAD will reduce us to give the NADH, FAD reduces to give FADH2, and FMN reduces to give FMNH2. So these are the forms of reduced coenzymes that are produced during the biological oxidation of the substrate molecules. So here I have used two words. One is called as oxidation and other one is called as reduction. So before going to the core concept of electron transport chain, we are supposed to well aware the core meaning of the two terms, that is oxidation and reduction. So firstly, what is the exact meaning of oxidation and what is the exact meaning of reduction? So if you consider say Fe2 plus, so iron is in 2 plus oxidation state, this Fe2 plus can lose one electron and thereby it get changes into Fe3 plus, and this Fe3 plus on receiving one electron will again convert back into Fe2 plus. So here Fe2 plus is losing the electron and Fe3 plus is gaining the electron. So here I used to say when a species loses the electron, that process is called as oxidation, and if a species gains the electron, that process is called as reduction. And if both the reactions, that is oxidation and reduction, if both are taking place simultaneously, we used to call that particular reaction as redox reaction. And in a system, if you are having both oxidized as well as the reduced form of the same material, such as Fe2 plus by Fe3 plus, here you can notice the material is iron, and iron is in both 2 plus as well as in 3 plus oxidation state, and they are present all together in such a way. Here you can notice for Fe2 plus, Fe3 plus is the oxidized form, and for Fe3 plus, Fe2 plus is the reduced form. So having together both oxidized and reduced form of the same material is actually called as redox couple. So one doubt may comes to our mind. Suppose if a material is given to you and if we ask, so which material will whether the material will undergo oxidation or reduction, how to judge, how to say whether a given material have the tendency to undergo oxidation or tendency to undergo reduction? In order to answer for that particular question, we need to go with the standard reduction potentials of the respective redox couples. I repeat, in order to account for the tendency of a material, strictly speaking, the redox couple, I mean to say a material, whether it undergo oxidation or reduction, we have to go with the standard reduction potential, in a simple abbreviated form, SRP values. So depending on or based on the SRP values, we can judge that a particular material will undergo oxidation or reduction. So in the field of biochemistry, we can apply the same concept and we can mention some redox couples. So for example, NAD and NADH, these two are the redox couples. For this, NADH is a reduced form, and for NADH, NAD is the oxidized form. So this is one redox couple. So for such a kind of redox couples, we can write the standard reduction potential values called as SRP values, and based on the SRP values we can say whether a particular redox couple is having the tendency to lose the electron or gain the electron. And now we shall look over into the standard SRP values of the redox couples of the reduced coenzymes. So for the first redox couple, that is NAD and NADH, for this redox couple, the standard reduction potential, that is E naught value, is minus 0.32 volts. And for the second redox couple, that is FMN and FMNH2, for this redox couple, the standard reduction potential is 0.30 volt. And for the other redox couple, FAD, FADH2, the standard reduction potential value is around 0.22 volts. So hope you can notice, so all these three redox couples are having negative reduction potentials. And now we shall consider a few more examples for redox potentials for the respective materials. So for the material coenzyme Q, which is called as cubiquinone, called ubiquinone. Q is ubiquinone. And for this coenzyme, the standard reduction potential is 0.10 volt. And for cytochrome C, the standard reduction potential is 0.25 volts. And for the other cytochrome, that is a combination of cytochrome A plus A3, the standard reduction potential is 0.29 volts. And for the reaction, that is for half O2 H2O. For this, the standard reduction potential is plus 0.82 volts. So you can notice here we have written 4 redox couples here and 3 redox couples over here. So you can differentiate these two sets, that is these 3 redox couples are having negative standard reduction potential, and these 4 redox couples or materials are having positive SRP values. Remember, the material which is having a negative electrode potential or having the tendency or will show a tendency to lose the electrons, means the materials that are having negative electrode potentials are the good electron donors. Similarly, the materials that are having a positive SRP values or positive reduction potentials are the good electron acceptors. So these materials can readily undergo oxidation, and these materials can readily undergo reduction because they are the electron donors and they are the electron acceptors. So by looking over to this particular data about the SRP values and the meaning of oxidation and reduction and how the thing will happen in case of metabolism, we shall try to learn the exact meaning of the term electron transport chain. So we said the coenzymes such as NAD, FAD and FMN are having the capability to donate the electrons to the electron carriers, because we have seen the standard reduction potentials of the redox couples containing FAD, NAD and FMN are negative in sign. So because of the negative standard reduction potential, we can easily say that these materials acts as a good electron donors or electron transferring agents. So in the cells, the electrons will get transfer from the reduced coenzymes to the electron carriers, and this process of transfer of electrons from the reduced coenzymes to the electron carriers that actually happens in a cell is collectively called as electron transport chain. And simply we can define the thing, that is electron transport chain, as the transfer of electrons from the reduced coenzyme to the electron carriers in the cell is collectively called as electron transport chain. And this electron transport chain is actually occurs in the inner membrane of mitochondria. The site of electron transport chain in a cell is the inner membrane of the mitochondria, will actually gives the provision for the electron transport. So if you consider the structure of the mitochondria, this is the outer membrane, and inside of the mitochondria there will be a structure like this. This is the inner membrane. So this is the outer membrane, this is the inner membrane of the mitochondria, and you can notice between the outer membrane and inner membrane there is the intermembrane space. There is a space between the outer membrane and inner membrane, that is called as intermembrane space. And in inner mitochondrial membrane, there are various components that are present in a definite order. They have been arranged in a definite sequence, and that helps in the transfer of electrons from the reduced coenzyme to the utmost final oxygen so as to produce the water. And the sequence of the components that are present in the inner mitochondrial membrane for the process of electron transfer will be like this. That is the sequence of components that are present in the inner mitochondrial membrane will be like in this sequence. Firstly, there will be NAD, which is a coenzyme, and first the electron will get transferred from NAD to the coenzyme Q, and from coenzyme Q to cytochrome B, and from cytochrome B to cytochrome C1, and from cytochrome C1 to cytochrome C, from cytochrome C to the complex cytochrome, that is A plus A3, and finally the electron will get transferred to oxygen so that the oxygen will get converts into the form of water. So this is the actual sequence through which the electron transfer will occur in inner mitochondrial membrane. And in inner mitochondrial membrane, that is in case of electron transport chain, for the easy transfer of electrons from the reduced coenzyme to oxygen, there are 4 complexes have been involved which are most important in the transfer of electron in the electron transport chain. And now we shall look over into the each and every type of complex that present in inner mitochondrial membrane which perform the transfer of electron from the reduced coenzyme to the oxygen. The complex 1 is called as NADH coenzyme Q, oxidoreductase is the complete name of the complex 1 that actually takes place in electron transport chain in the inner membrane of the mitochondria. And this complex 1 contains FMN, that is flavin mononucleotide, as a prosthetic group, and along with flavin mononucleotide, this complex 1 also contain iron sulfur proteins, and these iron sulfur proteins will not contain a heme group, and because of that reason these iron sulfur proteins are also called as non-heme proteins. Iron sulfur proteins are also called non-heme proteins. These proteins do not contain the heme group. So in complex 1, that is NADH coenzyme Q oxidoreductase, will be having FMN flavin mononucleotide as a prosthetic group, and iron sulfur proteins are also present, and because of absence of the heme group in these proteins, they are also called as non-heme proteins. And in this complex 1, the electron transfer will occur in a particular sequence, and now we shall see what is the sequence for the electron transfer. Complex 1, first the coenzyme NAD will receive the 2 protons from the substrate molecule so as to convert into the reduced form of coenzyme, that is NADH and 1H plus. So this will transfer electrons to flavin mononucleotide, that is to FMN, which is acting as a prosthetic group in complex 1, and that will get changes into FMNH2, and from FMNH2 the electron will get accepted by iron sulfur protein, that what we are calling as non-heme proteins, and from iron sulfur protein the electron will be transferred to the coenzyme Q, so which will get reduces to semi-quinone first, and once again get changes to quinone, which can be written in a simple abbreviated form as QH2, and this is the sequence of electron transfer that occur in case of complex 1. Okay. Complex 2 is also called as succinate dehydrogenase is the name of the complex 2, and the complex 2 is also called as succinate coenzyme Q reductase. These are the names that we can give for complex 2. And now we shall see which is the prosthetic group present in succinate dehydrogenase, that is in complex 2. So we said in case of complex 1, FMN will be present as prosthetic group, and in the second complex, that is the succinate dehydrogenase, the prosthetic group present is FAD, that is flavin adenine dinucleotide is present as prosthetic group in case of complex 2, that is succinate dehydrogenase. And the complex 2 is also contain iron sulfur protein like complex 1. And now we shall see the sequence of the electron transfer that actually occurs in case of complex 2. In complex 2, first from succinate, the electrons will get transferred to FAD by proton transfer. So FAD will get reduced to give FADH2, and from this FADH2 the electron will get transferred to iron sulfur protein, and from iron sulfur protein the electron will get transferred to coenzyme Q, that what we also said, that is ubiquinone. And by receiving the electron, coenzyme Q first will get changes into semiquinone, and this semiquinone will further get reduced to give quinone, that is QH2. So this is the sequence of reactions that occurs, sequence of electron transfer in case of complex 2. And now we shall see how the electron transfer will occur in case of complex 3. Next, complex 3. Complex 3 is also called as coenzyme Q cytochrome C oxidoreductase is the complete name of the complex 3. And this complex 3 also includes cytochrome B, cytochrome C1. So this is the other two cytochromes that are present in case of complex 3. So here, like in case of remaining complexes, that is in complex 1 and complex 2, complex 3 is also having iron sulfur proteins. So here, like in case of previous two complexes, we shall see the sequence of electron transfer that occurs in case of complex 3. So just you notice the name of the complex 3, coenzyme Q cytochrome C oxidoreductase. So these are the two extreme components that are present in case of electron transfer chain of the complex 3. So first, the coenzyme Q will get changes into semiquinone, that is here we are continuing the sequence of electron transfer from where the electron transfer has been ended in case of complex 1 and in case of complex 2. So in both the cases we have seen that the sequence have been stopped at the conversion of coenzyme Q finally into quinone. So first coenzyme Q will get changes into semiquinone, and this semiquinone will get reduced to quinone, that is QH2. So from that QH2, I'm writing the electron transfer directly from this. The actual thing that happens in case of coenzyme Q is first it will get changes into QH2, and from there the electron transfer will occurs to iron sulfur protein, and from iron sulfur protein the electron transfer will occurs to cytochrome B1, and from cytochrome B1 to cytochrome C1, and at last from cytochrome C1 to cytochrome C. So this is the sequence of electron transfer that occurs in case of complex 4. And now we shall see what's going to happen in case of complex 4 in case of electron transport chain. So the complex 4 is also called as cytochrome A plus A3 oxidase is the name of complex 4. So this complex 4 is also contain iron in both oxidized as well as in reduced form, and this cytochrome is also contain copper in both oxidized and reduced form. So we are having two redox couples in case of complex 4, that is cytochrome A plus A3 oxidase. Okay, so in case of complex 3, the electron transfer has reached the point of cytochrome C, and now the electron transfer will get continue in case of complex 4, that is from cytochrome C the electron will get transferred to cytochrome A plus A3, and from cytochrome A plus A3 the electron will ultimately get transferred to half O2, and from half O2, since the oxygen is electron negative in nature, once the electrons were received by the oxygen, it will get changes to O2 minus, and that will accept the two hydrogens or protons so as to convert into H2O, and thus oxygen is the final acceptor of electrons. One doubt may comes to our mind. So oxygen can receive electrons directly from NADH or directly from FAD or directly from FMN. So why should the electron transfer should occur in all these sequences, and finally it should come to cytochrome A plus 3, and then only why the electron should go to oxygen? The question may comes to our mind. The answer is, among all the different kinds of electron acceptors or electron carriers that are present in the system, cytochrome A plus 3 is the only electron carrier that can directly react with molecular oxygen. Not cytochrome C, or not coenzyme Q, or not iron sulfur proteins, or not cytochrome B1 or NAD or FAD. This is the only electron carrier that can directly react with molecular oxygen, and so that the electron get transferred from the cytochrome to oxygen so as to produce water. This is the reason why all these sequence of electron transfer has to occur, so that the electron should come to the cytochrome A plus 3, then the electron may get changed to or transferred to molecular oxygen so as to change into O2 minus. By receiving the two protons, it may get changed into water. So these are the sequence of electron transfers that occurs in the complexes that are present in the inner mitochondrial chain, that is strictly speaking in the electron transport chain. And now we shall draw a picture to show the complete sequence of electron transfers that actually occurs in the inner mitochondrial membrane by mentioning the four complexes and the sequence of electron transfer. A diagram for explaining the electron transport chain in inner mitochondrial membrane. So this is the membrane, the dotted lines what we have mentioned, and there are totally four complexes. This is complex 1, this is complex 2, this is complex 3, and this is complex 4. So in between of this there will be coenzyme Q and cytochrome C. So in first case, the hydrogens will be received by NAD 2 plus 2 electrons. So by receiving the hydrogens from the substrate molecule, NAD will get changes into NADH plus 1H plus, and from this the electron will get transferred in case of complex 1, that is to FMN, and from FMN the electron transfer will takes place to give FMNH2, and from FMNH2 the electrons will get transferred to iron sulfur protein, and from iron sulfur protein the electrons will be transferred to coenzyme Q, and from coenzyme Q the electrons will be transferred to complex 3, that is to iron sulfur protein, and from iron sulfur protein the electron transfer will be to cytochrome B, and from cytochrome B to cytochrome C1, and from cytochrome C1 to cytochrome C, and from cytochrome C the electron transfer will be to cytochrome A plus A3. And from cytochrome A plus A3, the electron transfer will take place to molecular oxygen, and the electron transfer will be, the magnitude of electron transfer is 2 electron, and by accepting 2 protons and 2 electrons, this molecular oxygen is finally changes to H2O. And the same thing we can notice with the complex 2. Here first the succinate will will transfer the electrons to FAD, and FAD will changes to FADH2, and FADH2 will transfer the electrons to iron sulfur protein, and from iron sulfur protein the electron will be transferred to coenzyme Q, and again the procedure is same. But one doubt may comes to your mind. So both complex 1 and complex 2 will transfer the electron to coenzyme Q, and thereafter the electron transfer will continue to complex 3, and there again to intermediate cytochrome C, and again to complex 4, and finally to molecular oxygen to produce water. Right. So in case of electron transfer from complex 1 as well as from complex 2, there occurs generation of energy, and that makes the pumping of protons into the intermembrane space, that is to the space which is present between the inner mitochondrial membrane and outer mitochondrial membrane. So in case of electron transfer from complex 1, the energy released will be equals to approximately 12 kilocalories, and that is enough for pumping the protons into intermembrane space. But again, if you come to the case of complex 2, the energy released during the electron transfer is very much less, and thereby the pumping of protons out of the membrane is not possible because the energy produced is not enough. And from complex 3, the energy released is around 10 kilocalories, and this is also having capability to pump the protons. And in case of complex 4, the energy released will be little less, and thereby compared to complex 1 and complex 3, complex 4 will pump relatively less number of protons. As a whole, complex 1 during its electron transfer will pump 4 protons into the intermembrane space, and complex 3 also pump 4 protons into the intermembrane space, and complex 4 will pump 2 protons into the intermembrane space. So as a whole, 4 plus 4 plus 2, a total of 10 protons are pumped into intermembrane space. And because of presence of these positively charged protons, due to presence of protons which are positively charged in the intermembrane space, the whole region of this intermembrane space will become potentially gradient, or you can clearly say due to presence of more number of protons in the intermembrane space, there creates some proton gradient across the membrane. That is, the outer membrane surface, outside the inner mitochondrial membrane, there will be more positive charge. Like this. And more negative charge inside the membrane. And because of this presence of proton gradient, the inner mitochondrial membrane is not possible. Because of presence of a positive charge across the membrane, the protons are not permeable and because of this positive charge present across the membrane, the inner mitochondrial membrane is impermeable to protons, so that the oxygen should receive the electrons from the cytochrome A plus 3 outside the membrane, and these protons will receive by the oxygen so as to produce water. And this is the electron transport that occurs in inner mitochondrial membrane, and that's the whole and sole thing about the mitochondrial electron transport chain.